A very good afternoon, one and all. I wholeheartedly welcome all the participants to second section of the day under the 6-day workshop on pilot plant scale-up techniques organized by Gabriel Hanumaya Institute of Pharmaceutical Sciences and sponsored by AICTE and ISTE. I, Dr. T. Balkrishna, Department of Pharmaceutics, take a privilege to introduce Dr. T. G. K. Murthy sir, speaker of this present section. Dr. T. Gopal Krishnamurthy sir completed bachelor's in pharmacy with a distinction from Gulbarga University in the year 1992 and master's in pharmacy from Birla Institute of Technology in the year 1995 with a GPA score of 3.87 out of 4. He was awarded with PhD in pharmaceutics from JNTU University for the title Studies on Ethyl Cellulose and Cellulose Acetate Films and Microcapsules, and Design and Evaluation of Controlled Release Microcapsules of Diclofenac from Andhra University in the year 2004. And he is currently working as a professor and principal at Bapatla College of Pharmacy, Bapatla, with a total experience of 25 years. Few of his outstanding achievements in the field of pharmacy includes a total of 258 research publications, out of which 113 Scopus indexed journals, and 4 patents are granted, and he also publishes 4 patents and 4 books published by him. He successfully guided 22 PhD scholars and received a AICTE funded research projects, and he also conducted various conferences. Total conferences conducted by him is 4 and grants received from various statutory bodies. And he also received Meritorious Teacher Award from JNTU for 3 times. And he also received Best Teacher Award from JNTU in the year 2019. It is a great pleasure to have such an esteemed person with us today, and I request him to take over the section.
Okay, thank you, Dr. Balakrishna, for your nice words and introduction. Am I audible?
Yes, sir, audible, sir.
Okay. So at the outset, I wish to thank the principal of the Gabriel Hanumaya Institute of Pharmaceutical Sciences, Dr. Vidyadharan, convener, Dr. Shashidhar, the faculty members of CHIPS, and the faculty members who joined this online mode. Very good afternoon to all of you. So today I wish to present a talk on scaling up by using QBD. So now this quality by design is one of the emerging area related to from starting formulation development, which includes from R&D to up to the stage of releasing the product into the market. So throughout this validation of various stages are required. So how to go for continuous validation and minimization of these validation steps, what are critical material attributes, critical process parameters, critical operation procedures, and critical quality attributes to be thoroughly discussed, then only one can get a knowledge on the factors, the features to be considered while addressing the product development in various stages. So now, whether QBD or this pivotal batches or scale up, is it a part of stage of development? Yes, it is one of the part in development of a pharmaceutical dosage form, which not only restricted to either solid dosage forms or liquid or semi-solid dosage forms, sterile or non-sterile dosage forms. So each and every pharmaceutical dosage form need a thorough investigation and knowledge on QBD aspects. So throughout the continuous validation process, it is required. Next, how this validation or QBD in pivotal batches is measured? It is measured with the success of robustness. If a product is having product or process. In both the aspects, robustness is required. So if you successfully developed a product with good robustness as well as the process which is used in development of a pharmaceutical dosage form, those processing conditions are having the scalability, then we can say yes, it is successfully developed. Next, whether any prior knowledge or first principles are required. So, whenever you want to develop a process, the scientific knowledge is required. Based on that scientific knowledge, we can identify the factors responsible for the product development. Why I am emphasizing this prior knowledge is, as the faculty members, the learned faculty members well know that the drugs can be classified into 4 categories under BCS class. The problems related to class 1 drug may be different with class 2 drugs and similarly class 3 to class 4. So as the problem is different, in order to address those problems, the solutions required, the strategies required, or the approaches required in order to pursue class 1 to class 2, class 2 to class 3 and class 4 are quite different. So in order to identify what are the material properties required which are influencing the final attribute may be different from class 1 to class 2, and hence the basic principles and prior knowledge is required in order to identify the fundamentals which are influencing the quality attributes of the final or finished dosage forms. That is why in order to screening of experiment or in order to limiting some of the factors, theoretically there may be n number of factors which may directly or indirectly influencing the final quality attributes. But we have to restrict them due to some practical or technical constraints. So which factor should be highlighted, which factor is more significant, that has to be thoroughly highlighted and investigated by using the knowledge of QBD and its interpretation. Next, when this aspect is required, at what point we have to apply this QBD. So as you know, there are different stages before launching the product into the market. The first one is selection. Selection includes the objective based on the literature development, literature review. So which product you want to prepare. So identification of the product that has to be highlighted and that should be selected. Next, we will go for development, R&D. So in R&D, normally there will be NDA or ANDA. In case of NDA, there will be a first time they have to develop, and in case of ANDA, that may be development of a generic product which is similar with already approved innovator product. Then we have to ensure that the product developed is similar or on par with the approved and innovator product. So what are the strategies required that is included in development stage? Next, exhibit batch, where we have to increase the size in order to tune the demand for clinical trials. After getting clinical trials data, then we can go for filing to various regulatory authorities. That may be CDSCO in India and FDA in case of USA. Later, this regulatory agency is going to review. If they are satisfied with the data submitted by the authorities, then they will give permission. Then it will be going for the launching of the product into the market. So in between the review and launching the product, there may be the need for pivotal batches of different sizes. So this good where we have to start this scale up is in between lab scale to pilot scale. That is the ideal place where we have to go for scale up techniques. So scale up technique is always better to be launched in between successfully developed R&D product and before going to the pilot scale. So preliminary requirement is there should be availability of successful process and successful formula. Then before going to larger scale, we have to go for scale up studies. And risk-based approach. So in QBD, we have to identify what is the risk involved in both the composition as well as the process, and what is this design space is before development of a pharmaceutical product, we have to set certain quality attributes that is called as dependent variables and our outcome variables. Say in case of a tablet dosage form, disintegration, dissolution, hardness, friability, these are all outcomes. And this outcome may be influenced by certain critical material attributes like particle size, physical form, and process parameters like binder addition and what is the RPM of that granulator. And these are all called as independent parameters or independent process parameters as well as independent composition parameters. And they are likely to influence the outcome. Say in case of a disintegration time, this disintegration time is quality attribute, and this may be influenced by critical material attribute like particle size. If the drug representing BCS class 2, if a drug representing BCS class 1, then particle size is nowhere influencing the disintegration time. So that is area where we require the fundamental or basic knowledge or principles of science involved in pharmaceutical dosage form development. Now the question will arise, which particle size is required to get the required disintegration time? Say 10 micron particle size is identified as critical. Means until unless the particle size is less than 10 micron, there is no required disintegration time is attained. Means above 10 micron, we should not use. At the same time, the particle size is also influencing hardness of the tablet. Normally, the particle size is less. The space between two particle sizes is less. Thereby, attractive forces or coercive forces are dominating, and hence the strength or hardness of the tablet is expected to be high. And friability. So, in this way, the same particle size, same parameter, same critical material attribute is influencing various outcomes of the product. So, which size to which size, it is influencing their outcome or the target that one has to identify. That is called as design space. So, within that space or window, that parameter is influencing the outcome that one has to identify. Next, design space verification. Okay, we identified the design space, perfect. Next, at the same time, whether it is practically verified or not. How to verify that space? Because some cases, the same parameter is influencing more than one output. One parameter may be influenced linearly or directly, and another parameter is indirectly. Means, if the particle size is reduced, the dissolution may be improved. So, for dissolution point of view, a low particle size is better. But at the same time, if you are reducing the particle size, it is badly affecting the micromeritics or rheological properties, the flow properties like Osner ratio, Kars index, tapered density, bulk density. So the number of granules occupying the die cavity during compression, these are all influenced. So low particle size is required for disintegration, hardness, friability, dissolution point of view. But low particle size is not beneficial for improving the flow property. Then we have to go for optimization or compromisation. At which particle size both the attributes, because we need flow as well as disintegration and dissolution. So at which particle size both the critical quality attributes are observed. That is the required, and that one has to verify in the design space. And scale up models. Why a scale up model is required? And this scale up model is nothing but establishment of a relationship between dependent and independent variables. Is there any relationship between those two variables or not? If there is existing a relationship between dependent and independent variables, whether it is a linear relation, like a simple equation, regression equation, y is equal to a plus bx. Or if it is not a linear relation, it may be sometimes a non-linear, that may be a cubical or quadratic equation. Then the equation such as y is equal to a plus bx plus cx square. So such kind of relations one has to establish And some cases, there may not be a simple relation. There may be a multiple regression analysis. Yes, the quality attribute may be influenced by more than one parameter, not only X. It may be X1, X2, X3, X4, X4, like n number of factors. Same Y may be influenced by X1 to Xn factors. And there is also cases where X1 influence not only Y1. X1 may influence Y1, Y2, Y3, and Yn. So what is the relationship existing between those parameters? Is there any possibility to develop an empirical equation to correlate the dependent and independent variables? That is nothing but a model. Some cases, in order to develop a model, computer simulation models may also be used nowadays, like artificial intelligence, machine learning, augmented reality, virtual reality. These are all areas where we can go for simulation. When we can go for simulation, simulation is possible only by knowing the relationship existing between the dependent and independent variables, at what conditions there is a strong relation, and what should be the data or what should be the environment to be created, under which environment there is a possible relation between dependent and independent variables. So in order to pursue, in order to identify, solve those issues, definitely there is a need to go for development of a models in scale-up. And what is the use of such kind of scale-up models is the effect of input parameter on output response can be precisely determined by developing the model and facilitate faster process optimization. As I mentioned earlier, X1, X2, X3. So normally one factor analysis, OFA. If you are doing OFA, it will take more time because initially we have to analyze one factor, followed by second factor, followed by third factor. Individually, one factor should be analyzed, followed by second parameter. But in our pharmaceutical process development, there will be n number of factors. So one by one, if you are solving its influence and optimization, it will take more time. So in order to avoid such kind of time-consuming process, it is advised to go for multivariable analysis. More of the variables involved, and all variables can be simultaneously solved by using MVA analysis, which is possible by multiple regression analysis and multiple ANOVA. Thereby process optimization can be expedited and provide a simplified version of a complex process. So this process is complex in pharmaceutical industry because it is influenced by n number of factors. And out of n number of factors, only some of the parameters may be practically significant. So which parameter is significant and which parameter is not significant, and which parameter can be excluded in our experimental design. Normally, it is advised to go for screening of the experiments initially. In screening of the experiments, the possible parameters and their influence on outcome will be thoroughly investigated, and the data will be subjected to ANOVA, and by default, it is set at 0.05 level. That indicates the hypothesis is correct at 95%. In that analysis, what is the p-value? Greater than 0.05 value will be neglected, and there is no need to include such parameters in further process. Thereby, this process can be simplified, splitted into the important parameters and non-important parameters. And understand the process robustness. Normally, the process as well as product both should be robust. Robust in the sense, even minute fluctuations in the process, exactly if the temperature to be maintained in the drying is 70 degrees centigrade, but it is not possible. There should be some allowances between 65 to 75 means plus or minus 5% like that. Then only we can say robust, because exact temperature, exact flow is not at all possible. So if the process is allowing some deviations at the set level, then we can set this process is robust, and we can identify from which temperature to what temperature this process can run smoothly so that it can be precisely controlled and make process predictions. So there will be inward direction. Inward direction means the flow will be continuously moved during process and product development. So that at what temperature, at what flow rate, what is the outcome? By developing the suitable empirical multi-regression analysis, one can predict what is the effect of input process variables, input process parameters, as well as the composition parameters. If you change that input, how output is affected can be easily calculated by using such kind of equations. That is why there is a need to go for scale-up models. And critical material attributes required to be optimized and controlled during high shear wet granulation process. So these actually critical material attributes are different from solid dosage form to liquid dosage form to semi-solid dosage form, and even in case of solid dosage form, tablet to capsule dosage form, powder to granule, and even in case of tablet also, from dispersible tablets to effervescent tablet. So as the type of tablet is changed, then also it is going to be varied, and it is also critical material attributes are also likely to be varied from one class of drug to another class of drug. For example, if a drug representing BCS class 1 and another drug is representing BCS class 2, then its material attributes are going to be changed. And in this slide, you can witness the critical material attributes pertaining to BCS class 2. Because the first point here which I am going to address is PSD, particle size distribution of API. And particle size distribution of API is not influencing the solubility and permeability of class 1 drug. It is not influenced by particle size, so there is no need to study the importance of particle size distribution on dissolution, disintegration, or any other parameters. Of course, in case of class 1, particle size distribution may have influenced the micrometallic properties, but the quality attributes, namely disintegration, dissolution, SA, drug content. So those compendial tests are not influenced by particle size distribution of class 1 drug. And hence, so if the drug belongs to BCS class 2, then at what size you get the required disintegration, dissolution, that one has to study, and that should be controlled, and one has to fix so and so particle size is required. Then one may question, so whether this particle size distribution is common for all BCS class 2 drug? No, it is not so. So for each and every drug, we have to change, we have to study the particle size distribution, though they belong to same BCS class 2 system. Next, the question will arise whether we scale up means, say we are having a formula of paracetamol tablet containing 500 mg strength of paracetamol. If you want to go for 500 mg paracetamol to 650 mg strength, is it called as scale up? Or scale up means initially we are having a formula for batch size of 100 tablets. Now we want to go for batch size of 1000 tablets. Then it is called as a scale up. So both are comes under scale up. Initially we are having a formula for 500 mg strength. Now we want to go for 650 mg strength. Then earlier formula may not work out. We have to go for scale up, both composition as well as process. Similarly, second case, if you increase the batch size from a batch of 100 to 1000 or 10,000, it is also considered as a scale up. So the second parameter is particle size distribution of excipients. So blend homogeneity is required in order to address the drug content issue and drug content uniformity issue in case of potent medicaments, and homogeneity of blending. And so this particle size distribution of excipients and particle size distribution of API, if both are same, then there is a possibility of homogeneity of blending, which is reflected by scale of scrutiny, scale of homogeneity, scale of uniformity, where you can get a good blend uniformity. And in order to address that, the particle size distribution of excipients is also required, and one has to conduct the required experiments in order to investigate influence of particle size and its distribution and its impact on various quality attributes of the dosage form. And the third question is polymorphic form of active pharmaceutical ingredients. So nowadays, most of the drugs which are currently available are either amorphous or crystalline form. And in case of crystalline form, they are available as polymorphic form. And my learned friends well know that the polymorphic form and amorphous forms are influencing the disintegration, dissolution, and bioavailability issues. So the formulator will show a keen interest to keep a particular polymorphic form for its identified bioavailability dissolution criterion. But during the presence of other components or during the processing, like even solvent, say in the literature, one might be using absolute ethanol. And second case, it may be consisting of hydrated alcohol, means alcohol and water mixture. Then the drug may be previously it may be in anhydrous form where there is no water. Now we are using as a granulating fluid alcohol water mixture. Then there is a possibility of conversion of anhydrous form to hydrate form, which is going to enhance, which is going to alter the disintegration, dissolution, and bioavailability aspects. So the solvent. Another one is the polymorphic form may also be observed during milling mixing conditions or even drying because temperature is applied. During the temperature, there is a possible conversion of one polymorphic form to another polymorphic form. So the formulator has to ensure that during product development, there is no changes in the polymorphic form. Whatever the polymorphic form initially used, the same may be maintained throughout the development of dosage form as well as throughout its life cycle up to its shelf life. It should be maintained in the same. So one has to identify the conditions which are involved in the polymorphic form and it should be made robust. And the fourth one is moisture content of active pharmaceutical ingredient. So some of the components are hygroscopic in nature, or some of may be deliquescent or efflorescent, which may absorb or lose moisture. And because of changes in the one minute. Okay, sorry for the inconvenience. So moisture content of APA. So moisture content generally anhydrous form is said to be more anhydrous form is said to have better disintegration, better dissolution. But during process, because of the influence of neighboring components or even packaging materials and processing conditions, the material may pick up moisture, thereby its solubility may be altered. So during scale up and process development, initially what is the moisture present and throughout the process, same moisture content is maintained or not. This concept is equally applicable not only API and excipients also. Because anhydrous lactose and spray-dried lactose, lactose monohydrate, these materials of flow properties are different as well as its effect on stability of the API as everybody knows Maillard reaction where lactose monohydrate is involved and anhydrous lactose is not influencing the stability. So during initially if you are having anhydrous lactose, but during processing, if it is converted to lactose monohydrate, then there is a possibility of chemical reactivity, and even moisture may catalyze some of the chemical reactions. So stability, thereby the drug compatibility issues may also be erased. Next, critical process parameters required to be optimized during high shear wet granulation process. So one is impeller mixer speed. What is the influence of mixer speed? Why it should be optimum? If it is less than or higher than optimum speed, what will happen? If it is like a ball mill, if it is a low speed, then there will be only cushioning and there won't be any mixing. And if it is having high speed, then the material will be thrown towards the walls of the casing. Thereby, again, the mixing is not going to be observed, and sometimes there may be aggregation and splitting of the particles and generation of very fine particles. And hence, mixer speed is one of the critical parameter, and it should be scaled whenever we are moving from lab scale to high speed. My intention is to clear that in case of a lab scale, if you are using 10 RPM for 100 tablets and 1000 tablets, what should be the RPM means? It should not be multiplied with 10. So simple mathematics should not be applied. For 10 tablets, this is the RPM, and 100 tablets, it is multiplication of 10. It is not correct. So one has to investigate whenever the speed is altered, what should be the lower case, and it is depending upon dimensions of the impeller. And similarly, the chopper granulator speed is another parameter which should be addressed. In case of chopper, granulator speed is, it is purpose is in order to reduce the aggregates into smaller particles. So like earlier here also, the chopper blade and its speed is critical, and it should be identified and well controlled in order to get the required particle size and required mixing homogeneity. And the third one, binder solution addition or spray rate. And what is not only the concentration of binder solution, and again this question will arise, which binder solution you want to add. And here it can be addressed as qualitative factors and quantitative factors. Qualitative factor means one is used PVP as binder and second one is HPMC as binder. So binders are different, both are used at same concentration. Even in case of PVP, one is used PVP K30 and another one is PVP K45 grade. Then what is the influence of PVP K30, PVP K45? Both are confined to same concentration. Then that is also the influence of binder solution effect. That is called as qualitative factor. And the second question is, you are confined to PVP K45, but one is used 2% and another one is used 5%. Then as the concentration of binder is used, obviously low friability value we will get and more hardness is get. But disintegration, dissolution are reversed. And one need a physically stable tablet which is reflected by low friability, and the quality attribute, another quality attribute is better dissolution, which is possible with low concentration. But as a formulator point of view, it requires a good physical stability as well as better dissolution. And hence, what concentration of binder we will get both required attributes. That is influenced by how much time you are using for incorporation of binder into the powder blend. So if you are taking more time, then that indicates you are using more concentration, and hence the quality attributes are likely to be altered. Next, wet kneading time. How much time you use it to form a damp mass. So how much time is used, and what is the effect of time? So if the time is more for mixing, then you are wasting the time, because in order to mix, for example, 5 minutes is sufficient. If you are allowing more than 5 minutes, that is not only wastage of power energy, it is also causing contact between the damp mass with the chopper speed, thereby it is going to be broken more, because there will be more contact time between the moving element and the damp mass, thereby there will be generation of fine. And similarly, dry milling rate. After drying, there will be, it is subjected to generation of the fine and uniform granules. So in order to get uniform granules, what should be the flow rate of the dam mass into the granulator, into the miller? So flow rate, because if it is more than the required feed rate, then the milling equipment may be blocked completely, because it is going to be dumped with beyond its capacity. And if the feed rate is less than the required, then wastage of energy. So feed rate means per unit period of time, how much quantity of wet granules are passed into the milling equipment, that has to be optimized. Drying temperature. And if the temperature is more than the required, then some cases the stability is going to be affected, because the chemical kinetics are altered with respect to temperature. And some cases the temperature is high, the granules may attain a moisture content less than EMC, and once the granules are withdrawn from the dryer, then there may be again absorption of moisture, so that is unnecessary wastage of energy. How much time it is subjected to drying. So it is some cases it is advised to go for high temperature and less drying time, rather than small low temperature and more drying time. So temperature and drying time both are interrelated, and they has to be simultaneously set. And once the granules are dried and get the required uniform size, then we have to go for compression. And during compression, how much compressional force is applied? Again, that range, the design space has to be set for compression force. High compression force may result, some cases, capping lamination problems, because there will be development of more stress, that is called as strain index. So strain index may be developed after that is called post compression. So after post compression, the granules will try to relax from the previously applied compressional force. In this connection, there is a possible generation of the stress or strain within the granules, and they may be exceeding the bond index. So in high stand indices, bond index, strain index, brittle fracture index, they are all influenced by the compressional force. And compressional force should also be accordingly, one has to set and one has to fix the range which should be preferred during compression process. So which areas one has to observe? So this is the process or flow diagram involved in tablet compression by wet granulation technology. And starting from weighing, next one granulation, that is formation of a dam mass. Then there will be wet milling, from wet milling to fluidized or tray dryer. And again, in case of dryer also, some cases in industry there may be using tray dryer, and some cases it may be a fluidized bed dryer, some cases a spray dryer, some cases a freeze dryer. Then the question may arise, if you are shifting from one dryer to another dryer, whether the quality attributes are changed. Because in our batch formula or lab formula, they may be used a tray dryer. Later, is it better to go for shifting from tray dryer to fluidized bed dryer? If we are going from tray dryer to fluidized bed dryer, is there any quality attributes are altered during scale up? Yes. In case of compared with the tray dryer, if you are moving for, if you are shifting from fluidized bed dryer, the size of the granules is going to be less. Because in tray dryer, the material is in static condition, and in case of fluidized bed dryer, the granules are in moving condition. So each and every particle will be having good flow property, and the need for glidant can also be reduced in case of fluidized bed dryer. And if you are using spray dryer, then the granules will be having spherical shape is issued, then more flow properties and better uniformity, and they are able to withstand good compressional force. And if you are using freeze dryer, then freeze dried material will be having high porosity, better dissolution. So the volume occupied by the granules is going to be less if it is dried to spray dried. So the dye cavity should be set accordingly. And if you are using a porous material, that is freeze dryer, then no solubility enhancers are required in the composition. and dry milling, followed by blending, where it is has to be mixed with some extragranular disintegrating agent, lubricating agent. Then how much amount of extragranular disintegrating agent, how much time it has to be mixed, and what should be the particle size of the lubricating agent, how much time it should be blended, that has to be thoroughly investigated and optimized. So whatever the dotted lines indicated in this slide are to be optimized. So except weighing and packing, the remaining operations has to be optimized during scale up if a drug belong to BCS class 2 and wet granulation technique is applied. Now, if you are observing in this slide, what are critical material attributes, critical process parameters, and how they are going to influence the granule properties, and which properties of granules are going to be affected by this critical material attribute and process parameters. So, one of the granule properties, particle size distribution. So eventually any formulator or any manufacturer will try to develop a granules which are having uniform size. So particle size distribution means mean and average particle size, means it should be having less scattering. Standard deviation or relative standard deviation from average particle size should be appreciably less. So in order to confirm that, always better to go for good size distribution. And this, in order to get good size distribution, which parameters related to process and which parameters related to material, one has to identify. So with respect to material point of view, particle size distribution and surface area and particle shape and morphology are going to be influencing. Because if the initial particle size of the active ingredient and excipients are not having uniform size, automatically the blend is differed with particle size and size distribution. So it is always advised to use the raw materials or input parameters in order to ensure that they are all having uniform size. And similarly, particle shape. Particle shape, if all particles are having similar shape, then the area occupied by those particles or surface area occupied by such particles can be predicted by using various mathematical equations depending upon the shape. If they are having different particle shapes, for example, some of the particles are having spherical shape and uniform, and some of the particles are having a needle shape. So these needle shaped particles can easily block between two spherical shaped particles, thereby the total volume, the total surface area occupied is going to be different, and hence the die cavity during compression may not be uniformly filled as the granules are not having uniform size and size distribution. Next one, the critical material attribute solubility. The solubility of the drug is going to influence the mechanical properties and strength of the granules. Because if the drug is having solubility in the binder solution can be easily penetrated into the material matrix, thereby cohesive forces or network or bridge is going to be formed in between the drug and binder. So the solubility is critical in order to indicate the porosity, surface area, strength, and other mechanical properties of the granules. And in case of critical process parameter point of view, spray rate. So how much time? So that is generally unit is ml per minute. So during a minute, how much volume of the binder solution is sprayed into the blend that is comprised with the drug and excipient. So that spray rate is going to influence the mechanical properties. If the spray rate is less than the required, then the binder incorporated is too less than the required, and the granule will be having less strength, and the spray rate is high, more binder is incorporated, thereby the granules will be having more strength and they can withstand the compressional force, but the resulting compact tablet may fail to meet disintegration dissolution criteria. Next, binder viscosity. So binder viscosity is influenced by two parameters. One is concentration of binder and other one is the solvent which is used. For example, polyvinyl pyrrolidine, it is prepared, 2% PVP is prepared, 2% PVP K45 is prepared in water and same 2% PVP K45 grade is prepared with IPA. Then, if you are observing the rheological properties or viscosity of same binder solution, same concentration, but the solvent used is different, then the viscosity is going to be different. So viscosity is going to be changed by solvent. Next, the same water you are using, but the PVP grade is varied or concentration of PVP is different. Then also the viscosity of binder solution is varied, thereby spreadability and the force required to spread that binder solution onto the blend is going to be changed, and we have to again go for scale up of the process, because once viscosity is changed, the process conditions required to induce the flow and spreadability are likely to be altered. So what should be the nozzle properties? What is the number of nozzles present and the distance between the spray gun to the nozzle? That is all going to be varied. And surface tension of the granulating fluid. So in some cases, in order to induce the solubility, dissolution rate of the tablet, there is a need to go for surfactant. And in order to improve wettability of the material, some cases the wetting agent may be incorporated along with the binding agent solution. So in the binding agent solution, if you are using surfactant, the surface tension of the binder solution is going to be reduced. Thereby, the chances of getting contact between the blend and binder solution is going to be improved due to reduction of contact angle from 90 to 45 or 45 to 0. Thereby, good contact, good wettability, good penetration, and more strength is going to be observed. And hence, the flowability of the granules, that is the process parameter, the flowability of the granules, which is reflected by either Osner ratio or Cox index, will get good, excellent flow by possible because of the how much amount of water is used, how much concentration of surfactant is used. And the next parameter, next critical material attribute is bulk density. And this bulk density is influencing the process parameter bubble fill. Bubble fill means the capacity of the container which is used to process either milling, drying, or mixing operation. Say, if you are using 1 kg volume, 1 kg of the blender, the material which is required to fill that is influenced by particle size. Normally, if the particle size is less, it is going to occupy more surface area and the quantity to be filled is going to be less. In controversy, in vice versa, means if you are using the particle size which is having large, then it is going to occupy more quantity in the blender. So the quantity of the material, how much amount of the material can be charged into the equipment used for various unit operations involved in the processing of pharmaceutical dosage form is influenced by bulk density and how much quantity of the material is filled into that. Water uptake capacity, contact angle, wettability, these are all mentioned in the discussion of surfactant and surface tension. And particle surface properties. So surface properties means some cases, the surface properties like generation of static electricity, whether they are having what is the surface charge present. This surface charge may induce some cohesive attractive forces between the similar and drug and excipient particles. So what should be the charge present? Because of those charge, aggregating tendency of the particles and even the quantity of the final moisture content remained in the finished granules is influenced by that parameter. So these are all various critical material attributes and which process parameters are influencing granule properties. These are all the basic fundamentals or basic principles one has to know before investigating its influence and QBD with respect to the quality attributes. So up to now, with respect to wet granulation, what are the critical material attributes and critical process parameters and what is their influence on granule properties were studied. Next, What are the finished product critical quality attributes? So some may be in process and some may be after processing the dosage form. One has to identify and one has to establish the goal for critical quality attribute. So in case of particle size distribution of the blend is one of the critical quality attribute. So we have to set. This is the final blend should have the particle size between this size to this size. And there should be a scientific and well written proof for that. Well written proof means scientific evidence means below that particle size and above that particle size, which quality attributes are affected, and that has to be justified. Simply, we cannot say state that this is the particle size required. So, what are the risk factor? Below that particle size, what is the risk? And above that particle size, what is the risk involved? And that risk is finally reflecting the quality attributes. Say, above this particle size, disintegration time is changed from so and so time to so and so time. That is undesirable. And in that way, one has to justify. Flow property, because the flow property should be uniform. Otherwise, during compression, there may be a bridging, rat hole, etc. So in order to prevent that, it should have the required flow properties, which may be calculated by identifying, by measuring bulk density and taproot density of the blend. By using those two, various parameters, flow indexes can be calculated. And third one is average SA and relative standard deviation of blend uniformity, whether the blend is homogeneous or not. Before compression, it's not only the mixing of the drug and excipient or granulating fluid, whether the resulting blend is uniform or not. Then we have to collect the samples from different regions, and from each sample the drug content should be estimated, and from all these regions what is the average drug content and what is the deviation from that, how much deviation is observed from that average value, that has to be considered. Moisture content of the blend. Moisture content of the blend is again influenced by initial process parameter and material attribute. If the either drug or excipient contain moisture, or during process, if more spray binder is sprayed more than the required, or the drying is inadequate, means whether it is conducted at less temperature or whether it is conducted at less time, then it may contain moisture content. So what is the moisture content finally required? And that should also be stated, and it should be justified. Below that moisture content means equilibrium moisture content of that granules should be fixed at some level. Below that EMC and above that EMC, what is the effect? And in order to ensure the same EMC, what processing conditions, what material attributes are to be required, that should be identified in the R&D, and it should be confirmed in the lab scales. Because initial R&D we may use one moisture content. Whether same moisture content, is it okay if you are grading, if you are upgrading that size from initial to final levels. Essay of the finished product. So essay of the finished product, as per pharmacopoeia, there will be some standards. The essay, if it is in the monograph, definitely there will be essay value, and it has to be, it should be satisfied. Then the question will arise. In the pharmacopoeia, the percent drug content is 95. Can you fix 94? No, it should not be less than that. Then the second question is, if the pharmacopoeial monograph specified 95%, can we fix 98%? Yes, because it is more than the required. 95% is the set, and you want in your in-house, in your pharma industry, if you want to go for 98%, it is more hard to maintain 98% drug content, and you have to strive a lot of efforts in order to control the process and formulation variables, in order to control the assay, and we have to control the degradation, impurity profiles, and all those we have to analyze. So whether impurity and RS profile, impurity and RS profiles are also changed with respect to change of excipients, with respect to change of process, and with respect to changes of processing conditions. If you change the drying temperature, then impurity profile may be changed. if you incorporate instead of one binding agent flow rate to another binding agent flow rate, even milling speed, if you're operating milling at higher speed, there may be generation of more heat and accordingly there is a possible degradation. Even we are shifting from one equipment to another equipment, then also there is a variations in the impurity and relative substances profiling. Physical attributes. These physical attributes includes the physical appearance, thickness, diameter. So such kind of attributes are changed with respect to the critical material attribute. So if it is so, then which critical material attribute and which what process parameter and what operating variables are influencing that attribute, hardness, thickness, friability, disintegration time, dissolution profile. So whatever the items listed in this slide, each and every item should be having a preset value. Preset value means disintegration time as per Indian Pharmacopoeia for uncoated tablet should not, it should be not more than 15 minutes. So we should not say simply it should not be more than 15 minutes or it should be less than 15 minutes. Exactly what disintegration time is required. We want consistency in the product, not only less than 15 minutes. So out of 6 tablets, one tablet disintegration time is 6 minutes, second one is 10 minutes, third one is 12, fourth one is 2 minutes, and fifth one is 4th minute, and sixth one is 13 minutes. Then we can say each and every tablet observed disintegration time is less than 15 minutes. But we cannot say they are passed the disintegration test. Individually, all are less than 15 minutes. But if you are taking average disintegration time and relative standard deviation of the disintegration time, it is not going to satisfy the required targeting, that is QTPP, quality target product profile. So it is not satisfied. And hence, always it is advised to fix the in-house specifications or in-house requirements. This is the range of disintegration time which we are going to allow. Then, in order to get that set goal, is required to be identified which critical process and material attributes are influencing that. Then we have to identify and analyze each and every parameter, and we have to keep in each and every parameter within the specifications in order to confirm the final product is satisfying our required goal or set objective. And this is the risk assessment, how we have to go for risk assessment. So here, critical quality attributes, I mentioned five. One is physical attribute that includes this hardness, diameter, thickness. Second one, assay. And third quality attribute is content uniformity. And fourth one is dissolution. Fifth one is disintegration time. Next, so some are showed with here red color, some are yellow and some are in green color. And green color indicating low and yellow is indicating the medium and high is indicating, red color is indicating high. High means it is influencing too much. So physical attribute, granulation. So granulation process, granulation conditions are influencing the physical attributes. And wet milling, it is having medium effect, and drying condition is also having medium effect, and milling is having high effect because of the milling, the granule size, shape are going to be influenced. And blending is having low effect on physical attributes, and compression force is having high. For example, if you are applying high compression force, obviously the thickness of the tablet is going to be less. And in this way, physical attribute is going to be changed. And if you are applying more compressional force, the hardness is going to be increased and friability value may be less. And that is why it is indicated as highly influenced. Assay. And in case of granulation, it is having low effect. Wet milling also low. Why this assay is not influenced by granulation, wet milling is that is depending upon the stability of the molecule. So here, this slide is indicating a molecule which is stable molecule and belong to BCS class 2. If I am shifting from highly problematic molecule with respect to stability, then in such case, granulation, this low may be replaced with medium or high. Because in case of a molecule, say aspirin, if you want to go for aspirin tablet by using wet granulation, then in case of granulation, I have to put high. Because in wet granulation, water is used as a granulating fluid and aspirin may likely to undergo hydrolysis. So whatever this table indicating, it is not a generalized table, and this is varied with respect to one drug to another drug, and one process to another process, and one equipment to another equipment, and even R&D model to lab, R&D model to pivotal model, or R&D model to lab scale. And whenever the equipment dimensions are changed, the risk is also going to be varied. So each and every, depending upon the nature of the API, depending upon the nature of the excipients used, depending upon the quantity of API present, quantity of excipients used, this risk is going to be different. One cannot show this type of table for all molecules, or one cannot generalize the things. Whenever we are changing the either composition or process or critical quality attributes, then definitely one has to analyze this risk parameter, and this risk table has to be generated. Once such kind of risk tables are generated, we can concentrate on especially which parameter is highly influencing, and which critical material attribute and which process variables, which operating variables should be controlled in order to convert that high risk to low risk. Actually, it is high risk, but we can convert that high risk to low risk. Then only we can satisfy the product. How to get that high to low risk is we have to specify material properties. So if you are using this material having particle size from so and so to so and so, definitely we will get a stability. Such kind of preliminary experimentations or scientific evidence is established, then risk is going to be minimized. So that is the need of risk assessment. So dissolution, as I mentioned, it is a class 2 drug, and hence in case of granulation conditions, milling conditions, because in case of milling conditions, particle size. So if the milling is yielding less particle size, there will be more dissolution. And that is why in case of dry milling, it is put as high, and similarly compressional force is also influencing dissolution, as high compressional force is not favorable for dissolution. So red color is indicated. So in that way, each and every case we have to analyze such kind of risk assessment. Now, critical process parameters with respect to high shear granulations. So one is with respect to dimensions. Dimensions means from lab scale to upper scale, we are moving. Then such kind of dimensions, if are changing, yes, it is going to have high impact. That is, chopper ratio to bevel are not going to be similar. Means the diameter of the chopper to diameter of the mixing vessel. Simply, if you are in case of lab scale, we are having 1 kg, and we want to go for large scale of 5 kg. Yes, but what is the ratio initially observed between the diameter of the chopper blade to vessel? Whether the same ratio is maintained if you are going for 1 kg miller to milling equipment to 5 kg milling equipment, that one has to address. Next one is high shear mixer load. Here it is also shown as high. Like previous slide, here also this red color, yellow or green color are not arbitrarily, or they are not standard. It has to be changed, it has to be investigated case by case, and one molecule to another molecule. So dosing rate, mixing speed, densification time. Densification time means it is nothing but the granulation time, how much time you are allowed to get a contact between the drug and excipient mixture. The chopper speed is having medium effect. So in order to go for a scale up, which parameter is having high risk and which parameter is low risk? Normally low risk means there is no need to go for studying for in case of scale up, because whatever initially said, the same you can follow. Only we have to concentrate on medium and high, and the degree of association, degree of concentration is different. In case of highly influencing parameter, more criticality, more critical zone is required, and more design space should be controlled. So now, as we are in teaching profession, in order to give some sort of interest, I showed here one cartoon associated between a discussion or dialogue between a student and faculty. And a student asked a question to the faculty. Student question is, I saw that you added the binder by dissolving it in 90 degrees centigrade. Can't it go into the powder? Because binder is wet granulation. As the discussion now, up to now, it is confined to wet granulation. So binder solution is maintained at 90 degrees centigrade, and at 90 degrees centigrade it is incorporated. So if you are changing 90 degrees centigrade to other temperatures where it is going to the powder, that is the question. Then the teacher answer is, according to literature, it might affect the bioavailability. That is the basic principle. How it is influencing the bioavailability is binder 90 degrees centigrade. If it is changed from 90 degrees centigrade to other temperature, what will happen is, in order to exhibit binder its viscosity, the temperature is required. Because binder, until and unless it is not evenly and uniformly dispersed in the selected solvent, it is not going to exhibit its full binding potential. So at what temperature, at what conditions, at what concentration it is going to exhibit its full binding potential. If you are shifting those conditions, then the quantity of binder and its penetration, because inadequate binder is going to enhance the dissolution, and more binder than the required is going to impede the dissolution. So that is why the processing conditions are important. And let us see the second question raised by a student. Student, well, we use cold loop and heating to 90 degrees centigrade. There might be some safety issues. Because if you are using high temperature, the safety issues, because the solvent may get evaporated, and those inhalation of vapor, and some cases the residual solvents, and some cases the whatever the vapor you are leaving into the environment, there may be OSHA regulations or pollution regulations. Thereby, there is a need to control as per safety issues. That is the question raised by the student. Then, what is the normal answer by the teacher is, you okay, good. If you are reducing the temperature than 90 degrees centigrade, then automatically it requires more time. Because as the temperature is reduced, so obviously it will require more time for drying, etc. Let us see another question. Student maybe use an homogenizer to speed up the things. So homogenizer is normally, the student question is correct, because whenever we are using homogenizer, the process of mixing can be expedited, and within a short period of time, the chances of mixing can be facilitated. Then how the teacher is replied for that question is, yes, it is correct. But we are going within 2 weeks. We are shifting from R&D to manufacturing the pilot batch or bioavailability subject batch. Then we will see practically whether homogenizer is, unfortunately, it is going to facilitate good mixing. But at the same time, we have to ensure that there should not be any stability, bioavailability problems. So your input, that is, in R&D, homogenizer is not used, but the student is suggesting to go for homogenizer. Yes, it may be correct, but it has to be checked practically in the subsequent batches. That is the reply made by the staff member with respect to the third question. Next, if you are shifting, the another most commonly used industrial technique in preparation of tablets is direct compression technique. And in direct compression technique, what are the properties to be considered is particle size distribution of API, moisture content of API, particle size distribution, bulk density, tapu density of excipients, moisture content of excipients, polymorphic form of the API. These material attributes, one has to investigate, one has to identify, and the same properties one has to be used. Whatever the particle size used in the development of means R&D development, the same should be used in pivotal batches as well as bio batches or exhibit batches. And with respect to critical process parameters required to be optimized and controlled during direct compression processes, order of addition. Because you are having not only API, there may be disintegrating agent, there may be direct compressible diluent, there may be a lubricating agent. So which material should be initially charged into the mixing vessel? Next, in order to effect the geometrical dilution, which component should be added next? And what quantity should be added, that has to be addressed, and blending speed, blending time, compression force, and compression turret speed. These are all the critical process parameters with respect to direct compression technique. So initially, which parameters we have established, and the same parameters we have to check once again in subsequent pivotal batches or exhibit batches. And if it is working okay, otherwise, the influence of those dimensions of that equipment, the influence of those material properties again should be rechecked. If it is varied, then once again there is a need to go for optimization. And in process and finished product critical quality attributes of direct compression technology, which includes is particle size distribution of the blend, flow properties, assay, physical attributes, and assay of finished product, impurities, RS profiling, hardness, thickness, friability, disintegration time, dissolution profile. So these are all various in-process parameters to be addressed in case of direct compression technique. So this slides up to now my presentation is confined to what parameters we have to control during R&D scale, and the same parameters we have to shift. Not only simply the aspirin 10 grams, and in case for 10 tablets, and for 100 tablets you should have a multiplication of 10 times of aspirin initial quantity. We have to identify what is its initial particle size, and the same particle size whether we are using in subsequent batches or not. Because if there is any changes in the particle size in R&D batch 2 next batches, definitely it is going to affect the critical quality attribute. So initially R&D scale itself we have to describe the material excipients and processing conditions, then only we can go for pivotal, exhibit or bioavailability batches. And in this connection, whatever the material attributes, process conditions, that has to be thoroughly documented, and same one has to mention. And if it is influencing critical quality attribute or not, and if it is influencing, what is the relation, and is it possible to develop any empirical models or mathematical equations or semi-empirical equations and models. If it is there, and we have to thoroughly develop that equation, and by using that equation only we have to control the input parameters so that we can get a consistency in the final product outcome. So that is the most fundamental requirement. So with this, I wish to conclude my talk on this. Thank you for organizers and my learned faculty members for your time, valuable time to spend to learn and to showcase your interest, commitment, dedication in order to update yourself in various aspects related to this QBD related aspects with respect to product development, pivotal batches, all those things. Right? So if you are having any questions, you can raise so that it can be clarified, so that it is an open discussion to all my learned faculty members. So open forum for questions.
The section is open for questions. If any participants ask questions, ask.
Dr. Balakrishna, if there is no questions, we can wind up.
Okay, sir. Okay, sir. Thank you, sir. Thank you for your wonderful section, sir. If no questions, I can close this section. Okay, sir. Thank you, sir. Thank you for your fruitful section.